Eddie, thank you for that introduction and for inviting me. It's really an honor for me to be here. I'm trained as a physician and as a scientist, and I thought for my 15 minutes I would just share some personal anecdotes and experiences that have framed the way I think about this. And I'll just preface it by saying, it's an incredibly exciting time. I mean, it's sort of the golden age, and a lot of patients have benefited, and I think obviously the question today is, can we ensure that the rest of the world can benefit from this technology? So I'm going to start with this slide, which is a slide that my colleagues and I at Sloan Kettering made almost 10 years ago, and we purposely titled it The Vision for Precision Cancer Medicine. And I'm not going to walk you through it, but you can see the vision is now reality. We take a blood test and a tumor biopsy, and we do sequencing. We do this on every single patient we see at Sloan Kettering, and the similar practices are in place at most of the major cancer centers in the US and many in Europe. And that information initially was purely research and interesting. Maybe it would guide eligibility for enrollment in a clinical trial. But today, this is a standard way of making a diagnosis and determining a treatment choice. So to frame the way I think about this, back to my, it wasn't that long ago when I was a postdoctoral fellow, there were still debates about what is the cause of cancer. We now know cancer is a disease of mutations. I was personally involved in the development of Abl kinase inhibitors in chronic myeloid leukemia, which is a dramatic success story, as you know. And I was right at the front row of EGFR inhibitors in lung cancer. And I'll show you those two examples briefly. But I think those two examples back in the early 2000s is what crystallized a concept that we thought might be true, but we really didn't know until we had these pharmacological tools and clinical experiments to show it. This concept of a driver mutation and a dependency of a cancer on that driver, hence the idea of targeted therapy. And I was also right in the front row at the planning meetings for the Cancer Genome Atlas Project, which basically took the examples from those two cases and a few others and said, we can't just sit by the wayside and let these discoveries happen through serendipity. We need to take an organized approach and see how many other cancer drivers there are. You all know the outcome, both through the US effort and the international effort. Hugely successful example of, I think, a big science investment that continues to pay off. It's also, in the bottom point there, it's the driver of an enormous financial investment by the biopharma industry, which, of course, has positive and negative implications, as I'm sure we'll get to in terms of drug pricing, in terms of the discussion. But I have actually lost count. I can't find a good accounting of the number of new cancer drugs approved since imatinib and Herceptin 20 years ago. It's clearly in the 100 range and likely even more, and it just keeps coming. So I thought really just to give you a sense of how far we've come, this diagram was literally the diagram that when I was a postdoctoral fellow that we were using to study the BCR-ABL translocation in chronic myeloid leukemia. And then I also want to emphasize that it wasn't that long ago, just 22 years, that the clinical trial that I participated in was one of the very first examples of a targeted therapy that not only worked, but it worked in essentially every single patient on the clinical trial. We now, that is still an exceptional outcome, but it's not a ridiculous dream anymore. We know, we can understand these drivers, and we can predict with some confidence if we have the right drug and select the right patient that there'll be a response. I thought I would just share one, clinical sample, example. This is a patient with advanced stage chronic myeloid leukemia, essentially worse stage than acute myeloid leukemia that Ali was just talking about. These patients, when I was taking care of them, would have about 2 to 3 months life expectancy. This is a patient who was in that situation, and 4 weeks after taking this pill once a day, you don't have to be a hematologist to look at the difference between the bone marrow sample on the right, which is completely normal, versus the monotonous full of leukemia sample on the left. And the other amazing thing was in that same 4 weeks, The molecular test of the day, which was a cytogenetic analysis, the Philadelphia chromosome in that box, the short 22, is completely gone. This was really a career-changing moment for me. Of course, every targeted therapy, whether it be an infectious disease therapy, a COVID vaccine, whatever, there's a loophole. There will be escape variants, and this is exactly the case with these targeted therapies. Here's an example of one that we discovered. But the silver lining is that the playbook is pretty clear. We can anticipate this, and now there's now 6 Abl kinase inhibitors approved for clinical use. This most recent one, called the Seminiv, is very exciting to me because it works by a completely different mechanism, and so-called allosteric mechanism, and when given in combination with the first generation ones, leads to the absence of any resistance in preclinical models. It's now an approved drug. It'll be being studied in combination. I thought I'd just remind you again how we got to where we are in lung cancer. And again, the pace at which this has occurred, I want to underscore how fast it's been. So this is a timeline of just, from 2003 to the present, in which we've divided lung cancer, which was one, sort of giant category, into all these different slices of pie. And the bottom slice of pie, every single one of those slices, there's now an approved effective therapy for each of the individuals who has that subtype. Again, we have resistance to those therapies, but again, we've overcome resistance. I think the best example of this is a drug called osimertinib, which not only did it address a resistance mutation that in the blue part of the pie chart accounted for literally half of the patients who relapsed on the first line EGFR therapies for lung cancer. It's now moved to frontline therapy. It has fewer side effects than the original drugs. It's really a spectacular example of rational, clinically discovery of mutation profiles from clinical trials driving the drug development process to make even better and better medicines. So as we all now appreciate, cancer is not tens of diseases, it's hundreds of diseases, and they are smaller in number, and that in order to diagnose them accurately, that cartoon of precision medicine diagnosis is now a reality. We need these kinds of tests, not just the genetic tests, but also the kinds of tests that Ali was talking about as well, that account for expression, proteomics, et cetera. Now, I think it's fairly obvious that as we divide cancer into all these smaller subsets, it becomes more and more challenging to conduct the clinical trials. First of all, we have to identify the patients because they are almost by definition rare. But as you'll see, I put rare in quotes because rare is not rare when you count them all together in one basket. It counts for 80% of all cancer. But the execution of clinical trials is the challenge. We can overcome it, but there's still more to do, and I'm happy to discuss that. We've already heard in the morning session the importance of cancer centers, and as I'll show you, of consortia of cancer centers that can help through data sharing paint the path forward. These are very much possible, I know from my own experience, but they're also quite challenging. And the last point on this slide is that in order to build those knowledge bases from these consortia, we're taking advantage of sequencing data and clinical outcome data from the patients we've treated at our cancer centers. But we have to remember that the generalizability of those insights clearly is generalizable to the patients we see in our cancer centers. But I think, as we all know, our cancer centers in the US and in Europe are heavily enriched for European ancestry patients and not for others. And it's not at all clear that the conclusions we make from this kind of data will be generalizable. Okay, so from that dream of a precision medicine test, I'll just show you that here's the test that we have. It's an example of one of many that are now commercially available. It's complex. There's nearly 500 genes in the test. That's chosen for all kinds of reasons, hinted at by Ali in terms of, comprehension, but also feasible. and sensitive enough not to miss an important mutation. The interpretation of the results of these tests is another issue. I think it's very solvable. AI is another component to that solution. But what's clearly important is we need the knowledge of what happens to the patients with these rare mutations. Here's an example of a slide that, that Ali showed something similar, in which I'm showing you from the MSK data the frequency of different mutations seen in this is now tens of thousands of patients. And the point I want to make is this concept called the long tail. So there's, a handful, 10 or 15 mutations that are present on the far left and account for that big peak. But this long tail is not just a collection of rare patients who we will never get to. This is 85% of all the cancer mutations. It's a very large number of patients, and so it's one that we have to acknowledge. And I think the current clinical trial system captures some of this tail, the sort of the tail on the left-hand side, because we can execute those trials and because the biopharma industry has the economic incentive to develop drugs for those patients. But the ones on the far right are a little more challenging. I'll conclude with an example of data sharing. So 7 or 8 years ago, when I was honored to serve as the president of the American Association of Cancer Research, an American organization that is very international in its membership and scope, we decided to try this experiment of sharing data, both clinical data and genomic data. And again, I'm happy to share some of the stumbles that we went through and some of the arguments against it. But quite frankly, through force of will, a bit of a bully pulpit, and a little bit of money, we were able to get it done. And now there are 18 cancer centers. We're expanding to add 4 more centers this year. They are mostly in the US, but we include Canada and 4 centers in Europe, and one of the new sites will be in Korea. And we've pooled the results from over 120,000 patients who've been sequenced at the cancer centers in these different institutions. All of that data is accessible in an open source way, all of the tumor data, and we've invested very heavily in partnership with the biopharma community to now get all of the clinical annotation that goes with all of those patients, which is a much more challenging task than collecting all the genomic data, which is structured and easy to port. As clinicians in the audience know, much of the clinical outcome data is unstructured notes, even though they're in electronic medical records, and even though we have natural languaging, NLP methods, it's still quite a challenge. And I'll just finish with a citation of a paper that was published just a couple of months ago, in which a screenshot of just the opening landing page of the Genie website is there. And you can't see it, but in that red box is over 120,000 patients, and that's the different mutation profiles. And I encourage you to go look it up, and answering a few questions showing, that promising that you won't try to re-identify patients, you're allowed access. So, thank you.